Before we jump in and begin talking about transcriptomes, I want to do a quick review of genome. That's a very widely used word. Even non-technical, non-biology people know what the term genome is, at least generally, but I find often there are some misconceptions or maybe better shortcomings in our understanding of even graduate biology students. So, the human genome is a representation of all of the DNA found in each of our cells. Now, other than a few random cells that don't have a nucleus and our gametes, which of course have half of our overall genome, all of our cells have 2 copies of our genome, plus mitochondrial genes. And often there are many different copies of the mitochondrial genome, depending on how many mitochondria there are in the cell. However, when we use the term genome, we basically simplify this. A genome is usually represented by the haploid size of the DNA in a cell. So basically, because humans are diploids, our genome size, or what is more commonly called the C value, is just 1/2 of each of our chromosomes. And the reason we do that is because largely those chromosomes are very much the same. We're going to have some allelic variation. There might even be some random mutations that pop up after many cell cycles because of mutations. But largely a single representation or a single line from the beginning of chromosome 1 all the way to the end of chromosome number 23 would represent pretty much the entire genome for both chromosomes, again, with those slight differences in allelic variations and maybe some polymorphisms. Okay? And so our C value for the human genome is about 3.2 billion nucleotides, A's, T's, C's, and G's. So if we line them all up end to end, there'd be right around 3.2 billion. Of that, the mitochondria is a very small, and about 24,000, I believe it varies some, but usually around 18 to 24,000 base pairs are vertebrate mitochondria. So it's mitochondria, although very important, is only a tiny fraction of our overall genome. Now, of course, the genome for genetic males versus genetic females is going to be different, right? Where females will have 2 copies of the X chromosome and males will have an X and a Y chromosome, and that Y chromosome doesn't have a lot of functional genes on it. So technically, if we're going to look at the entire genome or the C value, a male's is a little bit bigger because to represent it, you would need to show both the X chromosome and the Y chromosome. But for females, although the net amount of DNA in their cells is bigger, they have 2, again, largely identical copies of the X chromosome. Okay, so 3.7 billion base pairs for the human genome. So what would happen if we compared 2 randomly chosen people and we just looked at their genomes? So we sequenced their entire genomes, assembled them, made sure we had everything correctly, and then compared it. We'll talk a little bit in the future about sequencing and assembling. Well, if you did that, you would see differences. You would see allelic variants. You would see polymorphisms like we've got pictured over here. This is a single nucleotide polymorphism, very likely has minimal or maybe even no impact on the phenotype, but it's a real genetic difference. And then also you would be able to identify any major mutations. And this might be particularly important if you're looking at the genome of somebody who's suffering from some sort of unknown genetic condition, right? You could sequence their genome and see if there were any large discrepancies between their genome and the average run-of-the-mill genome. So there's some really valuable things. And of course, all of our population genetics and data for comparing between species comes from these comparisons. Many of the early data sets for phylogenies for comparing between species were not entire genomes, but they were small sections that were chosen based on their utility for phylogenetics. And so these differences represent mutational events in the past that have been passed down, and their frequency or their presence or absence are going to represent different things. And phylogenetics is just one way to analyze those. Population genetics and looking at an overall picture of the diversity is another way. And so that's where the data comes from. And today, these genome sequencing projects are usually the best way to gather data, especially large amounts of data, for phylogenetics, for population genetic material, for just about any project you want. Largely, next generation sequencing and genome sequencing has taken the place of targeted sequencing. Okay? So once we have sequence, we need to be able to assemble it. So I'm going to give you a quick overview of assembly in the context of a genome, and then we will compare and contrast it when we get to our transcriptomes. So basically, what we do when we are sequencing a genome is called shotgun sequencing. For reasons that we won't get into here, it's actually cheaper and faster to do random little chunks of the genome and just do a whole bunch of them and then put them all together using a computational algorithm. We put them all together like a massive jigsaw with many overlapping pieces. And that is genome assembly. Okay? Now, because these little tiny pieces are sequencing randomly, are sequenced randomly, what that means is we're going to have some parts of the genome way overrepresented. So if this represents the genome we're looking at, we might have a part that's got 1, 2, 3, 4, 5 different sequences that all cover the area that we're looking at. We might have another area where we only have 1 or 2 sequences that cover that area. Okay? So realize we get a lot of redundancy, but again, it's just faster and cheaper to do this shotgun approach and then put everything together at the end. So as we are assembling these pieces into larger areas, again, some with lots of overlap, some with little overlap, we call that a contig. So contig represents a fairly large piece of the genome, but by no means near an entire chromosome. And we put those pieces together. And sometimes there might be gaps because of random sequencing. We might have small areas that we have missed that just were not sampled by our random sampling strategy. And so we put together these contigs into a scaffold, and a scaffold represents a very large segment of a chromosome, or maybe an entire chromosome. And there might be gaps. We might even know about how big a gap is, particularly if we have a reference genome that we could align it to. So for instance, if you're sequencing a human genome, you already have many human genomes available, and we know basically how big it is, what to expect, and we could very easily line up our new genome with an older established genome and figure out what we're missing in the middle. Now, if we're doing a brand new genome that has never been sequenced before, we could in theory compare it to a closely related species, but even then there are often major rearrangements and differences. So often, once we get to the scaffold part, we might have gaps or we don't know, and we might not even know unless we've done a chromosome count, right? We might not even know if that gap represents a few DNA base pairs that we're missing, hundreds or thousands of DNA base pairs that we're missing, or maybe even 2 parts of different chromosomes. Okay, so the scaffold is our beginning to understand and do that. And eventually, as we get better and better coverage, or particularly if we have already established genome to map it to, we get a very good idea, maybe with a few gaps in our entire genome, but we get a very good idea of the sequence of the genome. And so we map our scaffold onto a reference genome. Mapping can also refer to identifying loci, genes, or other areas of interest on our chromosome. But often that process is more commonly referred to as annotating. So annotating means identifying any polymorphisms, identifying genes, maybe even some information about what those genes are, how many different allelic variants they are. So mapping is basically filling in all of the details of a genome. So you should be familiar with those 4 terms and know how they refer to a genome. Some of them also refer to a transcriptome. So we'll look at those here in a little bit. So to finish up, because of this random sampling strategy that we take when we sequence genomes, how much coverage do I need to represent the entire genome? So for instance, if the human genome, we're sequencing the human genome, which is 3.2 billion base pairs long, if I only sequence 3.2 billion sequences because of the overlap, I'm going to be missing a huge amount of the human genome, probably more than 50%. Right? Maybe on average, I'll target about 50% of it, a little bit more, a little bit less, if I just were to sequence 3.2 billion randomly chosen base pairs. So I'd still be missing a very, very significant percentage of it. So if I doubled that, you say, oh, if you got 50% the first time or near to it, then just do it again, and you'll have 50%. But if I did double coverage, again, because it's randomly sampled, then my next time I would have roughly 75%, a little more, a little less, right? Because I would, again, have a 50-50 chance, but I'd also have a 50% chance of resequencing things I'd already sequenced. So if I had 3x coverage, then I would have somewhere in the neighborhood of, I think it's whatever, it'd be like 87.5% on average covered. And so every time I re-sequence the genome, I'm going to increase my novel sequences, things that were never sequenced before. But particularly as I do it over and over and over again, I'm often going to be just resequencing things that were randomly sequenced and chosen the first time. And so, again, it's cheaper and faster still to do this way. But typically, on average, to ensure that you have 99.9 some odd percent coverage of the genome, we typically want to sequence at a depth of about 50x. And what that means is 50 times whatever the haploid genome size is. So if I'm sequencing the human genome, I would want, about 156 billion base pairs randomly sequenced. And when I assembled them, I would have only a few small gaps. I'd have 99.9% of the entire genome covered. Okay? And that's a very good general rule of thumbs if you want a little bit more coverage. But again, if you do 100x, you're only going to get a very few small percentage. So there's kind of diminishing returns as you add more and more sequence coverage to your genome sequencing project. And 50x is kind of industry standard. That guarantees that you're going to get a very, very large percentage, although not all of it. And then if you really, really need to fill in those gaps, there are more labor-intensive, expensive ways that you can go in and fill in those gaps by targeting those areas rather than just randomly sequencing. Okay? So let's move on to transcriptomes. Transcriptome sequencing is, in big kind of large terms, identical to genome sequencing. But there are some specific differences that are important for our understanding of it. Okay? So if we are sequencing a genome, our starting material is DNA. And hopefully we can start with many, many copies, which you can do for most species. You can grow a culture of bacteria and you've got many copies of the bacterial genome. Or you can take a liver sample from a human or blood or others. And depending on the tissue, usually have hundreds, maybe even thousands of cells that are represented in that sample. Now, there are also ways to artificially amplify the amount of starting material. Okay, but with genomes, no matter how you start, how much tissue you have, you're always starting with DNA. However, when we're doing a transcriptome, as the name implies, we are starting with the things that are transcribed from the genome. Okay, and so with the transcriptome, our starting material is RNA. Now, in cells, there are three types of RNA. We've got ribosomal RNA, transfer RNA, and messenger RNA. Here they are represented by their abbreviation. And especially in metabolically active cells, a large percentage of the overall RNA in a cell is ribosomal RNA and transfer RNA. And these, although very important for accurate translation of genes into proteins, there's not a lot of genetic diversity and not a lot of importance there. They can be used, and there's some variance between species. They can be used for phylogenies, and rRNAs especially have been used for that purpose. Right? But if I'm looking at, say, a liver cell versus a brain cell, they're going to have the same rRNAs. There might be different levels. We might have more rRNAs in one cell versus another, but they're going to have the same ones, so it's not really interesting. So often when we're doing transcriptome sequencing projects, we use methods in the lab to reduce or maybe even completely eliminate the rRNAs and the tRNAs in our sample. And so that leaves us with messenger RNAs. These are the main target when we're doing transcriptome sequencing. And just for your reference, sometimes this process of sequencing a transcriptome is also referred to as RNA-seq. Like that. So just for your reference, if you see it, I'll just use transcriptome sequencing, but that's maybe a little bit more of a technical term for this process. Okay? Now, our RNA, tRNA, mRNA, all RNAs are not really very stable molecules. So the first step in generating a transcriptome is to take our RNA and turn it into DNA. Now, in most eukaryotes, this doesn't happen. It's kind of a one-way street. Once DNA is turned into RNA, by design, and that's how we maintain flexibility and adaptability and homeostasis in our cells, those RNAs, especially the mRNAs, are meant to be short-lived, and we don't turn them back into DNA. However, there are some viruses that do this, and so there's a number of sources of reverse transcriptases that we can synthesize and purify and use to turn these mRNAs into DNA. And so when that occurs, we call this, I'm going to add this to our slide here, we call this cDNA. which is RNA that has been reverse transcribed into DNA. And C stands for complementary because it's complementary to the protein coding region of the genome. Okay? So once we have our cDNA, at that point, basically it's very, very similar, in fact, in most cases identical to genome sequencing. And then we just simply sequence a whole bunch of them, randomly sequence a whole bunch of all of our different cDNAs, small chunks of them, and then we try to reassemble them. And so the transcriptome sequence assembly algorithmically is very similar to genome assembly, but realize there's going to be a different outcome. With the genome assembly, we're trying to do the haploid copy of our entire genome. So if everything works well and we have adequate coverage, we would have 23 different pieces, right? Maybe 24 if we're going to include the Y chromosome, and if we want to include the mitochondria, we'd have 25 different pieces. Most of them are hundreds of millions of base pairs long, and that would represent the entire genome. However, with the transcriptome, it's different because we are sequencing these mRNA transcripts, which are only the coding region. And in humans, the transcripts, let's take a step back, because I was going to say something that was not quite technically correct, and I'll try and make that distinction here in a little bit. But if we look at the human genome, the protein coding regions of the human genome only account for about 1.5% of the overall genome. So it's a very small fraction of the overall genome that we're actually looking at, even if we were to find every single copied gene. Now, the reason I stopped and backpedaled a little bit back there was because many of these genes can be expressed hundreds, dozens, even thousands of times. If we have a cell that's making 2 or 3 really important gene products and just pumping them out, there may be thousands of mRNAs that are available and being transcribed to make a huge variety of that product. So even though that one sequence only represents a tiny, tiny fraction of the overall genome, if it's being transcribed over and over and again, we may capture it many, many times. Okay? And that's important and is an important quality of transcriptomes that we'll talk about at the end of this lecture. But just realize that we might have some of these represented many, many times over. Okay? Whereas in the genome, you only have it ever represented twice, right? Of that part of the genome and then the haploid copy. And typically, even when we line up a genome, we don't represent both of them. We might say, hey, there's some allelic variation at this locus, or a single nucleotide polymorphism here. But we don't often put both genomes side by side for like both copies of chromosome 1, for instance, because it is largely the same with only some small differences. But transcriptomes are a little bit different. Now, once we have our transcriptome, we still have this process of needing to identify what gene each of these comes from. Now, we've greatly narrowed down the possibilities, right? We know that if our methodologies are right, that each of these assemblies represents a piece or an entire gene. And we might have pieces of genes because still there's sometimes gaps, especially if we didn't sequence to a very great depth. We still might be missing parts of many of these genes. Okay, but even if we just have a part of it, we could identify the gene. And we'll talk about that methodology and go over that in our next discussion. But we still have gene identification and annotation. And so there are pipelines and programs, because we might have tens of thousands of transcripts that we would like to identify. So there are pipelines and programs to identify all of them in our transcriptome. Or there are other ways, if we are just looking for some candidate genes, rather than trying to identify everything, we could just go back in and say, well, is gene X in this population, or gene Y? And we'll look at, we'll talk about and even do a little bit for one of the worksheets. We'll do some of that annotation. But again, largely identifying them is somewhat similar to identifying genomes, but again, we're only looking for the protein coding regions. Now, once we have our transcriptome assembled, we need to start asking these same questions, or maybe even in the process of doing this assembly. How much coverage do I need to represent the entire transcriptome? And this is a little bit of a trickier question. With the genome, we gave you a pretty good ballpark. It's okay, 50x more or less, and you'll have really good coverage. What about the transcriptome? Well, think about when we contrasted the transcriptome to the genome. Your first thought might be, oh, the transcriptome is only 1.5% of the overall genome. So obviously we need much less coverage, right? Instead of 50x genome size, maybe I could get away with a much smaller coverage based on the genome size and still represent all of the transcripts in a sample, right? But another thing is, remember that some transcripts may be there at very, very high concentration, and other transcripts might be at very low concentrations. So if I don't have adequate coverage, I'm going to, because it's random sampling, I'm going to over and over and over again sequence the ones that are there at high frequency, and I may miss other, maybe even important transcripts, but ones that are not at high frequency in the cell. Okay, so this is a little bit of a paradox, maybe a catch-22, because it's a very small portion of the genome, but I need to do more to make sure I capture something of the differential expression that is present in that gene. In fact, that's one of the very important things that we can do with the transcriptome, is that we can analyze it to say, oh, not only what genes are being expressed, but at what level are those genes being expressed. And that's very different than a genome, right? With a genome, we're just basically saying, let's sequence it enough so we can know exactly what the genome is, and then we know that every cell, more or less, has one copy of that genome, or two copies if it's a diploid organism, but one haploid copy of that genome in every single cell, right? And a brain cell's genome in an individual is the same as a liver cell in that same individual, barring any mutations that occurred, but largely, 99.99999% the same, okay? However, transcriptomes are different, right? So if I compared transcriptomes of two randomly chosen people, in some ways this is not even a valid question, right? Because you have many, many, many different unique transcriptomes at any one time in your body. What's going on in your liver, the genes that are being used, is very different than what's going on in your brain. So if I compared, even in the same individual, a brain transcriptome to a liver transcriptome, I'm going to have some genes, commonly referred to as housekeeping genes, that are going to be found in both of them. These are just kind of genes that are important for day-to-day cellular respiration and other critical functions. Then I'm going to have a large portion that are tissue-specific genes, right, that maybe are only ever used in nerve cells, like in the brain, and others that might be only used in liver cells, okay? So we're going to have a very different set of genes for those tissue-specific genes. And so transcriptomes tell us not only about the cell, but what that cell is doing, what type of cell it was, and not only that, maybe even what type of cell it was and what metabolically is going on in that cell. Right? So if I look at a liver cell after you've been fasting for 36 hours, I'm going to see a very different transcriptome than one right after you went down to the taco shop and had all you could eat tacos, and your liver is all metabolically active and ramped up to help you process and detoxify and do all the great things your liver does. So not only do we have differences between tissues, we have differences between the same tissue types, but under different metabolic stresses or metabolic activities, right? And then we could even potentially look at disease processes. What does a cancer skin cell, look like compared to a healthy skin cell? What genes are being overexpressed and underexpressed in cancer cells? So it's a really, really valuable tool. So not only do we have an idea of what genes are being used in one cell versus another, but at what levels. So maybe we have the same gene that's being used in a cancer cell as in a healthy skin cell, but we have 4 times as much of it that's being used. And so to do that, we would need to figure out relative amounts of each transcript in our sample. And that is called a differential gene expression. So this is our most powerful tool for being able to analyze transcriptomes. And it gives us a picture of not only what genes are present in each of our transcriptome samples, but at what level are they present? Are they there at a very small percentage of the overall transcripts, or are they at a relatively high level of transcripts? And so it gives us a very powerful tool for identifying many, many different critical biological questions, right? Individual A versus individual B, tissue A versus tissue B, tissue A at time point X versus tissue A at time point Y. Maybe even like developmental stages, right? What genes are being expressed at an early stage of embryo development, and what genes are being expressed 4 hours later in a little bit of a different stage of embryonic development. And because transcriptomes are so critical to the homeostasis, to the way cells are interacting with tissues around them, to what that cell is able to do, it's an incredibly powerful tool for examining what's going on in different cells.